So welcome everyone, and thank you for joining us to the launch of the Global Research Grant Programme in Europe for 2024. I'm going to start now. My name is Dr. Stella Child. I'm the Research and Grants Manager at GFI Europe, and I'll be talking through some of the programme details. I'm also going to be giving a quick introduction to GFI. Joining us today is three of our grantees from previous years' grant programs to talk through some of what they've achieved with their program grant and their experience of working with us. After that, we'll go into the funding priorities for this year's call, as well as how to apply, and then we'll have a Q&A session where you can ask questions. So many of you will know this, but GFI is an international nonprofit. We're dedicated to accelerating the shift to a sustainable, healthy, and just food system. So we have over 200 staff around the globe with three key departments. We cover corporate engagement, policy, and science and technology. So as part of our science and technology work, we run a global grant program, which is fully funded by philanthropy and dedicated to advancing the science of alternative proteins. So our grant program, some of the highlights of it to date. So we've awarded over $21 million to 118 research projects. This is a truly global program. We have grantees in 21 countries around the world. So the program is focused on addressing technical bottlenecks, unlocking additional research funding globally, and fostering collaborations which will expand the field. So that's GFI, and now I wanted to let you hear from some of our grantees in Europe who have and their experience of the program. So I'll stop sharing, and it's my pleasure to introduce Milena Ivanesivic, who comes to us from Connectomix Bio, and who will share some of the work that they've been doing.
Hi, thank you, Stella. I'll take over with sharing. Okay, slideshow. Is it on?
Yep.
Okay, perfect. Okay, thank you. Hi, everyone. Thanks for joining the webinar, and thanks for GFI Europe for organizing and inviting me to present today CX Bio research. My name is Milena Ivanesivic. I'm a co-founder of CX Bio, and we are 2023 grantees of GFI Research Grant Call. With our project converting organic side streams to microbial lipids. It's an open access techno-economic analysis and the lab scale proof of concept. My presentation will introduce CX Bio as a company. I will share more on motivation behind the project that we have. We have project overview, and also I'll share more of the outcomes of the project and our experience with working with GFI. About CX Bio, we are a service and a software provider for biotech, scale-up solutions focused on fermentation. We work with labs, startups, larger companies, and we offer techno-economic analysis and life cycle assessment, mainly focusing on microbial solutions, looking into microbial proteins, lipids, biofuels as well, and biosurfactants, and also focusing on alternative feedstocks and gas fermentation. We were founded in 2021 as Connectomix Bio. Since then, we rebranded into CX Bio. We are based in Luxembourg, and we have 5 core team members and 6 advisors, and we've done more than 15 projects so far. And we also had a pleasure to work not only with GFI, but also with the European Space Agency as well. Looking into motivation of our project, I mentioned in introduction that our project is looking into techno-economic assessment of converting organic wastes. And we do this through 3 intermediates, which are methane, biomethane, actually methanol, and hydrogen. This conversion actually allows us to decouple the food production from conventional agriculture, but it also allows us to showcase use of alternative feedstocks and to explore early on their techno-economics. Then This project is also leveraging on precedents that alternative feedstocks already have on the market. So for people who are in the fermentation space, who know that there are already commercial scale facilities using methane. We have Unibio, we have Calista in China and Russia are these facilities, but we also have a history of using methanol with ICI in the UK in the 70s. And for hydrogen, we have Solar Foods, who already has an approval in Singapore for their microbial protein. So there is a baseline for choosing these alternative feedstocks already. On the side of the product, lipids, and our choice to choose lipids, this is driven by the need of alternative proteins for functional and sustainable ingredients. But also with choosing lipids, we are addressing the general market need. So we are seeing certain supply chain challenges and competition between food, oils and fats, but also biofuel production. And there's a general growing demand in sustainable ingredients that goes beyond food production. How and why do we convert our organic matter? You can see here general process flow diagram of our project, and I will address first the in silico modeling that we are doing. So we are converting organic matter, I hope you can see my cursor, organic matter into an aerobic we're putting organic matter into anaerobic digestion, and we're getting the biogas, which can be upgraded to biomethane. This biomethane can be subsequently fermented as it is, but it can also be converted into methanol, hydrogen, and carbon dioxide. So essentially, we're using biomethane as an intermediate to these two other alternative feedstocks. The reason why we're doing this is that anaerobic digestion allows us to have a very standardized processing of our feedstock, upstream processing of our feedstock. It allows us to have non-volatile molecules for which fermentation doesn't constantly have to be readjusted based on their quality, but it also gives us a clean feedstocks that can be introduced in food production. Furthermore, with different feedstocks, we can have different organisms, and this allows us to actually expand the portfolio of products that we are considering. So briefly, you can see here in case of hydrogen troughs, but also in case of methanol route, we have lipid droplets, and we are looking into 3-acylglycerols as the main product. In route of methanol troughs, when we are using methane, we are targeting more phospholipids, which are conventionally known as lecithins in food industry. Nonetheless, all three routes will give lipids as the main products and protein-rich biomass as a co-product. To complement our modeling, we also have, in collaboration with San Diego State University, lab experiments where we are cultivating different microbes, changing different carbon feedstocks, mainly between methane and methanol, in order to get fatty acid profile analysis, which we can then compare to products that are already on the market in order to assess the market value of the products that we have in our modeling. And this is complementing and feeding into the model. In general, modeling is a stepwise process, and it starts with literature review and designing the process flow diagram. Then we need to select the equipment, scale the equipment in order to understand the capital expenditure of this particular production chain. We need to have the list of raw materials, the amount of utilities and labor put into such facility in order to assess operational costs. And these together with product and market analysis will inform us about the financial metrics of such facilities. Since these are relatively new processes, we are validating certain steps with industry. So for example, we consulted with a natural gas and biogas company in Germany called VNG on our upstream converting organic waste into biomethane. We also consulted with CMO in Canada called Assurance Center on our downstream processing to make sure that things are aligned. After that, we do a sensitivity analysis, which will highlight cost drivers and hotspots. And with this, we can go back and improve the model and improve the processes. This will all at the end be captured in an open access publication with open access model and certain resources that we've collected over time. So this project is still ongoing currently. In terms of products that we have, our three different routes give two different products, which I already hinted in the slide before. We have phospholipids, or known as lecithins. They're very good in emulsification due to the nature of the molecule. They're used in baking industry to contribute to dough elasticity. But primarily, when we talk about alternative proteins, they contribute to meat taste, and there are quite a bit of research showing that oxidation of fatty acids and phospholipids is responsible for this characteristic meaty taste that we have. On the other side, 3-acylglycerols, which are maybe more familiar to general public, they very much contribute to juiciness of alternative products. We use them as everyday cooking oil, but they also contribute to texture of different creams and sauces as well. So these different applications are driven by different nature of these molecules, and we need to take these into account when we are designing our production chain. So in order to look, when using methane, we are getting phospholipids, and when using methanol and hydrogen, we are getting 3-acylglycerols. And these different energy feedstocks have different states. One, we have a gas, we have liquid, and this will dictate the type of the reactors that we are using in our model, and subsequently dictate the cost of these particular pieces of equipment. Next, the products are located in different parts of the cell, and this has to be addressed in types of equipments that we are choosing to break down the cells, but also the types of fractions that we are actually processing in our downstream processing. So in case of phospholipids, which are building blocks of membranes, we're targeting membrane component of the bacteria. And in case of 3-acylglycerols, we are targeting lipid droplets that are actually in lipid bodies in the cytoplasm. Next, these molecules have different chemistries, and we are taking this into account when choosing solvents, for example. So phospholipids are polar, while 3-acylglycerols are non-polar, so choice of solvent will be influenced by the chemistry of these two molecules. And finally, the type of fermentation that we are using. So in order to get 3-acylglycerols, we have a familiar nitrogen depletion. 3-acylglycerols are actually storage molecules. Cells use them to store carbon when there's not enough nitrogen. So this will mean that we have two-stage system where we have growth and accumulation, so fat batch fermentation. While when we have phospholipids, since they are building blocks of membranes, we are looking into expanding the membrane system with this inner membrane structures, and this is done due to the intrinsic nature of methanotrophs, which are using the enzyme called methane monooxygenase. This particular type of PMMO is embedded in the membrane. So in order for methanotrophs to take methane and convert it later on and integrate it into the biomass, they need to expand on this protein in their cell, and for this protein they need more membrane. So playing with ratios of methane and oxygen will induce the formation of the protein, and with this expand the scaffold membrane, which we then extract as lecithins. So after understanding the chemistry and biology, we then go into first step of modeling. This is designing a process flow diagram. I will be showing just one route here, but in a nutshell, the steps are very similar between three different routes. Process flow diagram means aligning all the equipment pieces that we need to convert raw biomass into a final product. And general steps of the process flow diagrams are cell treatment, product purification, that is mainly solvent extraction. Then we take into account solvent recycling because of the unit economics. It contributes, if you're recycling solvent, you're simply using less fresh solvent, and this is driving the cost down, but you also have less cost related to water treatment. Then we have product drying, and finally co-product valorization. After this, we go to steps 2 and 3. We need to assess a capital cost and operational cost. Backbone of capital cost is equipment purchase cost. So we do this through CX Bioequipment database that we have collected over time with our experience, where we have different capacities relating to different costs of different pieces of equipment. Similarly, we look into operational cost. We understand the cost of raw materials, utilities, labor, in order to make a conclusion on the cost of production and minimum selling price. And then finally, we want to assess the profitability of these production chains. For that, we are analyzing fatty acid profiles. It's a 3-step process. We're analyzing fatty acid profiles on the products that are currently on the market. We compare them to the fatty acid profiles of the samples that we have and see if there are similarities. And then based on this, we inform our model what is the market price of the product that we have. And then we can compare if this is sufficient enough compared to the cost of production to make this facility profitable. And these are the general steps that we do for all 3 routes. And we will be combining this into an open access publication with the open access model. And this concludes the part on talking about the project. I also want to share our experience with working with GFI, which has been wonderful, going from the application process, which is very straightforward. It also allowed us to have a wonderful collaboration with institutions all around the world. We had a wonderful support with media attractions. With this, special thanks go to Conrad from GFI Europe and Stella as well. These are just some of the articles that came up regarding our project. We had a chance to talk to a lot of people from the industry to discuss price parity, environmental benefits, and also lastly, but not the least, we have had a wonderful support during the project from fermentation team and Lucas and Adam who are leading the fermentation grantees. And then just the conclusion, big thanks go to the CX Bio team. This is the hard work of many people, and then with our wonderful collaborators that you can see on the left, Marina from SDSU, Sumesh from DTU, Subin and Sati from Verschuren Center. And if you want to stay tuned, please do follow us on LinkedIn and check out our website, or just contact me directly if you have any questions. Thank you.
Thanks so much, Milena. And I'm sure I speak for everyone. It's really great to see so much commitment to like open access resources that the whole field would benefit from. That's one of the things that we really emphasize in our program, although it is still, and I'll talk about this a bit more later, like possible, we encourage industry applications, and it's possible to protect your IP at the same time as making these sort of results of your grant publicly available. I'm going to pass now to our second grantees in Europe who were working on a cultivated meat project. So that is Dr. Federico Ferreira and Diana Marques, who will be sharing a bit more about their work. That's perfect. We can see your slides.
Okay, so hello everyone. My name is Diana. I'm part of Algae to Fish. This project was funded by GFI back in 2021. It was a 2-year project and it's already concluded. We brought you here today our motivations and our main and relevant results. And Federico, you can. So as Stella was saying, our project is focused on the cultivated pillar of GFI. And in our case, it's actually not about meat, it's about fish. And specifically with our knowledge here in Lisbon, in our institute, what we focused the project on was 3D scaffolding, and we basically used additive factoring techniques as bioprinting and electrospinning, and our goal was to produce the first fish fillet in this field. The Algae2Fish project had 5 objectives. We are talking a bit about all of them today. The first one was actually cell expansion, differentiation, and characterization, as this was not yet done before. And we used an available fish cell line that is the lax cells. And then for the scaffolding part, what we did was a library of biowinks, and we divided into myogenic biowinks that would have fish myoblast encapsulated, and then a library as well of fat biowinks. In each case, the biowinks were formulated with edible plant-based materials and also some cues, as for example, microalgae. On our third objective, what we did was the formulation of electrospun edible fibers. And then we tried to combine both techniques. And with this, we actually mimic the muscle fat part of the fish, and then the myosepta of the fish, that is the connective tissue that resembles more like nanofibers. In a final step, we also explored how electrical stimulation can be applied to this field and its impacts on muscle cells and tissue maturation.
Thank you very much, Diana. So Diana gave us an introduction of the project. So it is really a multiplex, a multi-vector approach. We use electrospinning, we use the cells, we use the printing, and we use electrical conductivity. So we select, we are not presenting all the results, we select a few results to show you. And the very first results that are very important has to do with the cells themselves. So are these cells able to proliferate, and in which conditions they can actually proliferate? So the culture media still have FPS, so it's actually a challenge for the community that is watching us today to get better media. So hopefully that some of you will be working on this. But with the media, that is the standard media for this fish cell culture, we are able to get different cell numbers and different duplication times, and we figured out that most probably, considering the fall increase and duplication time, the small duplication times, we are better if you start by sitting a smaller number of cells, 10,000 to 20,000, rather than 40,000, 60,000. So considering these two conditions, we use the galactose-based media, that is L15, but we also try a glucose-based media, and we figure out that regardless the type of carbon source that we add, it's fine. Both the cells can grow in both of them. So it's a false idea that we need galactose for certain fish cells, at least for this one. And concerning the duplication, 20,000 cells per centimeter square is a good seeding. The cells increase nicely, and we can actually work on this seeding to get cells enough for them to do our bioprinting and our differentiation protocols. I'm not sharing the differentiation protocols here for the sake of time, but what I can say here is that another challenge that the field has is good biomarkers that can actually tell us if the cell is an embryonic stem cell, if it is a muscle progenitor, an adipocyte, myoblast, and so on. So it's important also to invest in this type of vectors. But our focus of this project, it was really 3D manufacturing, and for that we are being very fortunately, because one of our master students that now is a PhD student, Afonso Gurmão, built a printer from scratch. So he decided he's a mechanical engineer, and we give him the challenge to make a printer that was able to do a dual extrusion. We need one needle for the adipose ink, and we need a needle for the muscle ink, that we could control temperature, because some of the inks that we use, the gelification is temperature dependent. We put the UV curing. We are not using the UV curing because of this for food applications, but it's still there in case you want to use it later on. Our very first prototypes were actually done by combining the two bioinks, but we didn't use the bioprinting. We use molding. So we can use bioprinting or we can use molding, and you can see the sushi type prototypes. Don't get fooled. This is actually the real salmon, the wild salmon, while these are the ones that we build with our BioInks. The idea of making these prototypes, it was to confirm that our inks have a nice behavior in terms of solidification, in terms of printability, cell survival, and so on. The second prototypes are already printed. So here we have the scheme of this printing will happen. So we imagine design. You can imagine how your food will look like. So actually, you could select from a menu where you can have different configurations, different architectures. So if you have a chef that is cooking and he wants to be creative, he can do whatever configuration he wants. Then we have to inform the computer about that design. The computer has to translate that design in something that the bioprinting understands, and then the bioprinting follows the instructions, and layer by layer we'll print first. In this particular case, is printing first the muscle bioink or a more pink bioink, and then later on you'll fill the gap for the second bioink. He does this one time, so that will be the first layer. Then he goes back and he do a second layer until he gets some thickness of the fillet that he's building. You can see that success was not attained at the first time. So the very first combinations of BioX and fat were not very nice, and you really have to change the setups, the parameters in which you print, making them slower and making the ink a bit more viscous in a way that you can get something that has a nicer structure. So to show that it is an iterative process and needs to be built. And when we did this, we start to be happy. We are finally printing something that looks like a fish fillet, but we are at the same time more ambitious, because the thickness of this that I'm presenting is a very thin thickness. It's like a carpaccio. It's not really a fillet. So we decided to design something that looks more a fillet with a certain thickness. So our ambition, it was to build something with 2 centimeters thick in terms of height, with 6 centimeters by 33, so that in the plate looks like a small piece of a fillet. We decided to use 2 BioInks to provide color and to provide shape. So let's say that if you want to mimic a whitish part and then an orange part, and we design it like this. And again, we will do this layer by layer. We'll be using first a nozzle for what we call the myogenic BioInk, then a nozzle for the adipose BioInk, and so on and so forth. We could also have electrospinning, like I'll speak about this a little later, on these interfaces. So, but here we have a design with different combinations, different propulsions of adipose BioInk versus muscle BioInk to make thinner or thicker white stripes, and you can see the results with thinner and bigger white stripes. But it will be interesting to see how this actually works. You can see your printer that is working, and you can see that is a bit less trivial than people think on first instance, because you have to, if you want to print a slope, our bioprinter has to accompany that slope. So you actually have to be sure that the control of the printer goes in this situation. We are not printing flat. The BioInk has to accompany that slope. And here you have another one. I'm going to put all them to run in a way that you can see them at the same time. So here is at the beginning. Here you already have a second layer. And here we see the second layer making the white part, the fat part as well. And in this way you can actually see the printing doing. We can improve our printing by this homemade printer that is still under construction. We have more ambitions for that as well. Let me now move slides. So apologize. So in this slide you have apologize. In this slide you have the stage by stage process in which we have the first layer, then you already have 2 layers, you already have 3 layers, and here you can see the details. Stop in the middle to see the details of the printing. So we are fairly happy with this prototype, but that prototype didn't combine still electrospinning with bioprinting. So we went further and we decided to combine electrospinning with bioprinting. So this is examples of the fillets, already combining them. So to do that, we actually have to put together in our bioprinting to replace one of the nozzles where it used to be a BioInk with the material that comes from a syringe pump and then for the ones of you that are not familiar with leather spinning, is a process in which we apply 20 to 40 kilovolts between the needle and the plate, and by doing that you can pull the material in fibers that are at the nanoscale, so thin as collagen. And by doing that you can do a very thin structure here that is very similar to the myoceptum. That is that part that separates the different regions that are a bit more muscle and more fat, and the myoceptum has a more thin structure on the middle. So we did that. What I have to say is that before that we have to develop edible electrospun fibers, and that the problem with the edible electrospun fibers is that it's not only the material that we use. Like here we are using zinc, protein from corn or maize, but the problem is that the solvents that we use also have to be not toxic. So we have to use acetic acid and ethanol acid. We are still working on this because in a way that we are able to obtain a consistent, we still have a bit of gelatin, and that's animal driven, and now you are moving on to remove the gelatin and replace by other plant-based proteins, and we are using an algae that has the flavor and the smell of the ocean to give nice sensorial properties also on this material. Here you can see the final prototype. So here is without fibers, and here is with fibers. The difference is thin, but is significant. That will make the difference in terms of the aspect and properties, and also the optical properties due to the algae. Here you can see the fillet, how it looks like. Is getting something much more similar to what it is a wild salmon. So the way that the consumer can actually enjoy the meal. The flavors and the smells are also already aligned with something that is pleasant and nice and fresh. And here I'm zooming in, doing a scan electron microscopy to show you the interface between the fiber region and the bioink region in a way to see that they are really coupled. So it's not something that you pick up and then will dismantle if you don't do it properly. And now I want to move to something completely different, that is the muscles of the fish are controlled by electricity. So we talked that we could help the maturation of the muscles by applying electrical stimuli, and to see if the cells will actually get better in terms of the number of nuclei by cell. It's one of the few cells on nature that has more than one nuclei by cell, that is the muscle cell, and you can see, and you did here the results. You use alternative current or pulsed current, that is depend of the regime that we are using, and we actually can see that if you don't have electrical stimulation, the number of nuclei by cell is lower. AC seems better than pulsed. Also the cell length, if you want a myofiber, you want cells that are long, are better, and you don't sacrifice do it, you don't want a very thin cell. So we are quite happy with the results, meaning that we can use electricity as a way to maturate the myogenics of the muscle. I forgot to say that in this particular case, we are not any longer using the CBAS cells, but we are using the macro cells. These cells have been developed by David Kaplan group with Michael Said as a PhD student, and he kindly allowed us to use these myoblast cells to make these experiments. So I think that in terms of impact, I just select a few of the results. I want to say that the impact of GFI and of this project was tremendous in our ecosystem. It was tremendous for our research group because kick off a new line of research that came after, to be motivated by Diana, that started a master on this topic, and after the master we got this project. And after that, we got the national project to carry on the research. We got a scholarship to support the PhD of Diana, that is now in the beginning of her second year. And we are also coordinating an European project that is FISTS in this sector, in which we get 30, we are not alone there, we have 36 partners. So it's important. But also at the local level, and here are the Clean Fish, that is the national project. At the local, another colleague of ours that is not here with us today, that is Carlos Rodrigues, also got the second GFI project to develop the media and micro carriers. That project is still running, and here is FISTS. But what I want to say is that is not only the outputs in terms of money, funding, and people working, it is the engagement of a strong community that loves what is doing, is passionate about that, and that actually can bring us a better future on the field of alternative proteins. That community, we can see it in many different levels. One of the levels is the Cell Agri Portugal, in which we get our conference last September, and it was 150 people. Many of them came from abroad, from different companies. It was also something that I will pass the word to Diana, because she will be much better to speak about that.
Yeah, so GFI allow us to create a really good environment here in Portugal, and as a student, also after this project, and actually during the project, I got my PhD in novel strategies to produce cultured fish fillet. So we are still, and we created a new line of research here in this topic. And regarding the association, is very good because we brought together all the researchers in Portugal that are motivated to develop this field. And the conference was our first event, and it was really great to see everyone together. And we got around 150 participants. Was very great. And for me as well, more recently, we created here, I co-founded, and I am the co-president of the University of Lisbon Protein Project. That is also a big accomplishment. Because we are now promoting within all the university the alternative proteins, and yeah, I'm very happy about it.
Okay.
Here are some images of this impact, and besides all the projects and the new things and that we start and we are proud of, we also, with the Algae to Fish project, we got to go to several conferences. Was really great. We went to Helsinki last year, the Cellular Agriculture Year conference, another one here in Europe, the Maastricht one, and then you can see as well our conference and how it went. And of course, we also had the privilege to go to the Good Food Conference and to represent both the project and the Alt Protein project.
Here is our team, but better than the faces, we can listen them directly.
Federico, I think that the sound isn't coming through. I know we can see the subtitles.
Apologize, what shall I do?
I think that that's probably, given the time, we probably should stop there anyway. It's really great to see the whole team though.
Okay. We finished. We just want to finish by presenting our last slide, if that is possible. Okay.
Yeah, that's fine.
That is about the conference. Okay. So to invite everyone to come to Lisbon on October 24. Thank you very much. Thank you, and apologize the extra time.
No, you were perfect. Thank you so much. And it's very gratifying to hear GFI be thanked for such an amazing ecosystem, but really I think you've built such an amazing thing in Portugal, much of which is off the back of your own effort. Okay, so that's brings us to the end of hearing from the grantees. I am going to now talk through this year's RFP, so our call and the priorities that we're looking to fund this year. after which we'll have a Q&A session. If there are any questions for either me about the grant or the researchers that you've heard from today, we can take them then. So as you may know, every year our grant program focuses on different topics which are picked for the most impactful technological sort of areas that we can fund. This year we'll be funding up to $3.4 million across 3 topic areas. So that is upcycled plant proteins, next generation fermentation downstream processing, and hydrolysates for cultivated meat. So awards will be for up to 24 months and $250,000, which is approximately 230,000 euro, with additional funding available of up to $50,000 for projects that involve collaborations that bring researchers who are new to the alternative protein field. Funding priority A is upcycled plant proteins. So this topic aims to understand if we can reduce food loss and increase the value of the whole crop by using side stream byproducts to form valuable food ingredients. On the right here, you can see a few examples of volumes of protein-rich processing residues. These are large volumes, produced in large volumes in North America, and this image is taken from GFI's recent side stream analysis, which I'd encourage you to explore. So if the right byproducts and processes are used in plant-based foods, this could potentially improve the price and scalability while also reducing food loss. So what we're looking for in this topic area are proposals that focus on all 3 of the following things. So identifying promising side stream from a food manufacturing process, one which has preliminary evidence that it's viable as an alternative protein ingredient. We're also looking for proposals that plan to optimize raw material processing using emerging methods to improve the yield and quality of one or more ingredients. And we also would like volume and economic models for the optimized process. So while we believe that there are a variety of very valuable ways to approach side stream upcycling, here we're focusing on side stream from food manufacturing only and not outside industries, and specifically for creating protein ingredients. We want to ensure that projects funded through this program are set up for commercial success. And so we will require evaluations of the potential volumes of the side stream as well as economic evaluations. They don't necessarily need to be full forecasted production volume models or techno-economic assessments, but more sophisticated methods will likely be scored higher. And we're also looking for a strong understanding of potential production volumes, showing that the volumes are high enough to feasibly contribute as a food ingredient. As I mentioned, the recent GFI side stream analysis is a good starting point for understanding what type of volume and economic valuations are appropriate here. We also don't necessarily require an end product prototype for this subtopic. You do need to demonstrate the functionality of the side stream protein ingredients for plant-based foods, but you don't necessarily need to work that up into a full prototype or plan to. So what are we not looking for in this topic? We won't be considering proposals that focus on solutions using side streams from non-food industries or non-food safe ingredients, reagents, or processing technology. We also won't be funding solutions that attempt to use food side streams without a clear vision of application to produce plant-based ingredients, or projects that attempt to use food side streams that are currently produced at negligible volumes or without commercial potential. The other aspect is we won't be funding proposals that rely on AI approaches that don't come with rigorous experimental validation. So funding priority B, which is next generation fermentation downstream processing. This topic focuses on addressing the downstream processing only. So current precision fermentation downstream processing is a multi-stage unit operation system to separate cell biomass, extract proteins, concentrate, purify, and then dry the material. The increased number of unit operations increases the capital costs, energy and water consumption, as well as the loss between stages, which all impacts the downstream yield and unit economics. So DSP can account for a significant portion of operating costs as well as capital costs, depending on the amount of equipment needed. and this topic aims to provide solutions. So one proposed solution to reduce the costs and increase the sustainability of precision fermentation, DSP, would be to develop novel approaches for protein isolation and drying that improve the unit economics. So in an ideal world, this could be integrated one-step capture and purification with high throughput semi-continuous processing and low energy consumption. And that continuous or semi-continuous DSP would also enable or complement continuous upstream process implementation as well. So we think that there would be enabling benefits from this, from solving this problem. So what are we looking to fund in this proposal? We're looking to fund innovation in precision fermentation downstream processing, specifically for food. This means an optimized, adapted, or novel unit operation for DSP in the form of, it could be equipment, consumables, materials, or a hybrid biological sort of chemical engineering solution. But we're looking for a demonstration of the technology as it specifically relates to food protein DSP for fermentation-derived alternative meat proteins. So this can be for any protein that enhances the organoleptic properties of meat alternatives. It could be flavor, color, or structure enhancing proteins, for example, collagen or heme. Any demonstration that you plan to do should be with the relevant food protein. We'd also like to see outlined how this approach will significantly improve the unit economics over current DSP systems, such as a model of the electrical or water consumption per kilogram of isolated protein. We also would like to see the path to scaling the solution to a commercial scale bioprocess unit operations for food ingredient production. So with anything involving scaling, you should acknowledge the scaling risks and outline your mitigation strategies. Finally, what are we not looking for in this funding priority? So again, AI or machine learning approaches or screens that do not have substantial hypothesis-driven frameworks or underlying training data to produce physical, tangible deliverables will not be considered. Also, solutions focusing on higher value ingredients like growth factors, enzymes, or other food processing agents. This topic is really aimed about addressing alternative protein ingredients for final food formulation. So protein with a focus on meat alternatives. We'll also not be funding solutions that do not improve water or electrical consumption of DSP, solutions that don't use food-safe materials or reagents or otherwise incompatible with the realities of DSP, technologies that lack a pathway to commercial scale for food manufacturing, or technologies that are only effective for one protein class. So, for example, those that would only work with casein or iron-binding proteins. Or that are directed only towards dairy and egg proteins. And finally, funding priority C, hydrolysates for cultivated meat. So this funding topic aims to address the fact that the bulk of cellular biomass in proliferating cells today is made from amino acids, and amino acids used to feed the cells come primarily from individual fermentation processes, only some of which are sufficiently scaled. So hydrolysates are a potential solution for this. They are where high protein raw materials are broken down by acid or alkaline pH, heat, enzymes, or fermentation processes. Hydrolysates can contain a mixture of peptides, amino acids, minerals, carbohydrates, and lipids, and they can have a large impact on bringing down the cost of cell media and thus the cost of cultivated meat and seafood. Proposed advances in hydrolysate for cultivated meat may fall along various processing stages, including the raw material selection and preparation, hydrolysis, the heat application, filtration, purification, or end product characterization. So we're really interested in solutions anywhere along that chain. And there are some examples of interesting open questions that may be appropriate for a proposal here. And you can see that they cover everything from raw material selection through to the parameters of the hydrolysis, the nutritional or and growth inhibitory factors in the hydrolysate and the batch variability, as well as downstream filtration, purification, and characterization. So any of these would be appropriate for a proposal. However, successful proposals must articulate the raw materials for testing and a justification for why they were selected, ideally matched to the relevant cell type and the specific metabolic requirements of that species. You also need to include a hypothesis for how the proposed solution will enable lower cost and higher performing hydrolysis ingredients, and the experimental approach for testing the performance of the various hydrolysates in cells relevant to cultivated meat manufacturing. So for this, I'd really encourage that you look up GFI's cell line development report, which came out recently, which includes the cells and species of highest interest. We'd also like articulated controls and comparators that would be used and any limitations and uncertainty in the devised approach. And again, the cost, energy use, and resource utilization, how it could be improved or be affected compared to existing practices. So what are we not looking for in this proposal? We won't consider proposals that do not have at least one primary aim of improving the processing or the characterization of hydrolysates, or proposals that use any materials or processing inputs that could be expected to cause food safety or other regulatory issues. We will allow proposals that include screening existing off-the-shelf hydrolysate performance in food-relevant cell lines, but it cannot be the sole focus of the proposal. And finally, the use of any animal-derived raw materials as a source for hydrolysates is only acceptable, animal serum use is only acceptable in media formulations if if serum-free formulations are not available, or if using the hydrolysate assists in advancing the development of serum-free media. So that covers the three funding priorities that we're soliciting proposals for in this year's grant program. And I'm going to quickly run over how to apply. There's far more details on this in our RFP, which is available at gfi.org/research grants. And there's a detailed FAQ there too. So I would encourage you to explore that. In order to apply, you create a proposal through our portal at grants.gfi.org. You set up a user profile there if you haven't already. And again, the details of what you need to include, the sections and the word limits, the length limits are all in the RFP document. There is a pre-review proposal screening. So if the proposals must adhere to the guidelines, meet the topic inclusion criteria, and be scientifically and methodologically sound in order to progress to the review phases. So I'd really encourage you to pay attention to the clearly outlined requirements of each of the topics, as well as the general length and proposal requirements. So while proposals are written in English, we recognize that requirement may mean that researchers are writing in a non-native language. And I want to emphasize that we evaluate on scientific merit, not on language proficiency. A reminder that the deadline for this year's request for proposals is May the 23rd. We won't accept proposals after the deadline, but if you have submitted before then, you can access and edit up till that deadline. And another reminder that proposals must adhere to the stated page limits, and that if they don't, they will be excluded from the review process. One thing I haven't mentioned here, which is around IP, is that we have different IP terms, which you can read about in more detail in the RFP document. So we have standard terms for both industry and academic applicants, and they vary. But generally, we don't prevent protection of IP, but we would expect publication of the results, but there is a delay period if you wish to secure IP. I'm going to stop sharing there, and if there are any questions, we have a Q&A function that I'd encourage you to use. I can see one question so far, whether a PhD graduate can apply for this. And the answer is yes, we accept applications from applicants that are at any stage of their career. If you are a trainee or an early career researcher, we'd expect there might be a supporting PI on the proposal. that you build the whole team with that in mind. But yeah, you can find more details about exactly what you'd need to demonstrate in the RFP, which I'm going to drop in the chat right now. I'm not seeing any other questions. Does anyone have anything that they haven't had the opportunity to ask? I'll give us another minute. And yeah, I'd really encourage you to, depending on which priority topic, to explore the resources that are available on GFI's website. So for priority A, to look at the GFI side streams report. For priority C, to look into the cell line development report. You should find all of those linked from the RFP as relevant resources in the topic sections. As we've only got a few more minutes remaining, I'll just take another opportunity to thank our panelists today for coming and presenting their research. I hope you found that as interesting and fascinating as I did, and really kind of amazing to see the impact that's come out of like a 2-year grant in both cases. So thank you, Diana, Milena, Federico, for attending, and I will let everybody go now. And good luck. I hope that your proposal applications go well.
Bye.
Bye, everybody.